Okay, in today's lab, we are going to look at the blood characteristics of a number of different animals and see what those blood characteristics can tell us about the lifestyle of the animals. Now, I plan to do this or record this lab in our actual lab on campus, but circumstances being what they are, things have changed a little bit, and I'm going to have to adjust and do this in my private underground lab here at an undisclosed location. So we're going to be working with blood of different animals today. So, of course, first things first, protective gear. So let's get that on. Okay, so we have eye protection, we got hand protection, we've got gloves on. Our clothes are protected, so we're good to go. All right. So what we're going to do today is look at blood from a bunch of different animals. And here's our samples. Okay, we have blood from a horse, a cow, a sheep, and a rabbit. Wait, no. Horse, sheep, cow, hold on. All right, that's a sheep. Cow, all right, that's good. Horse, sheep, cow, and rabbit. So we're going to run some different tests on these blood samples and see what those blood parameters tell us about lifestyle. We're also going to get a blood sample from a seal. Now, normally I would get this blood from Mystic Aquarium, from one of their captive animals, but again, given the pandemic, we have to adapt. But fortunately, I do have a seal right here, and I'll demonstrate how we can get blood from this animal and use it in our experiment. Seals have a blood sinus at the base of their flippers. This is a little group of blood vessels where you can inject a needle at the base of the flippers and easily extract a blood sample. Well, unfortunately, this blood draw, that's not really enough blood for what we need to do. So I've got another way to get some blood from a seal, so let's try that instead. Oh, all right. I think that's going to work. All right, so now we can start analyzing the... Hey, get your feet off the desk. So now that we have our blood, the first thing we're going to do is measure the hematocrit, or the percentage of the blood that's made up of red blood cells. And for this, we are going to get some blood into a small capillary tube and spin it down in the centrifuge, and then you can read the hematocrit directly off of a hematocrit reader card. And for this part, I'm going to turn it over to my assistant, Megan, who is going to walk you through this part.
Hi, I'm Megan.
Hi, Megan.
Today we're going to read a PCV in total solids. I already have my blood sample, and I've got it here in a lavender top tube, so it's already anticoagulated. So the hematocrit tubes I'm going to use have no additive in them, and they come in this blue container here. So I need to make sure that my sample is mixed in the container. And then I'm going to fill my PCV tubes about 2/3 of the way to the top. It's a pretty good amount in there. And then I'm going to put them in the clay here. You want to fill 2 tubes so that you have a counterbalance in your centrifuge. Maybe a little too much blood in there. Okay. I'm going to take both of these, place them in my hematocrit centrifuge. I have balances on the other side here. And then I'm going to spin them for 3 minutes. Okay, these have finished spinning in the centrifuge, so I'm going to take my tubes out. And you can see that the centrifuge has spun down, so now I have a clear distinction between my red blood cells here at the bottom, and then there's a clear layer of plasma here on the top. So I'm going to take this hematocrit reader card. What you want to do is line up the bottom of the blood line with the 0 line here, and you want to take the top of the plasma line with the top line up here. get those set. And then where your blood meets your plasma, that's what your packed cell volume is. And in this dog, it is 51%. So his packed cell volume is 51%. That's at the high end of normal. Normal is about 35 to 50%. And that's it.
Thanks, Megan. But shouldn't you be wearing gloves? Amateur. So now we can do our hemoglobin analysis. And for this, we're going to use spectrophotometry. So spectrophotometry looks at color differences between different solutions. We've taken our hemoglobin, added some Drabkin's reagent to it, which is based on cyanide. So you should not lick your fingers more than once or twice when you're doing this. The spectrophotometer will now measure how much light is passing through our samples. And from the color difference between those two, the amount of light that's either transmitted or absorbed, it can tell us how much hemoglobin is in our solution. So I know probably most of you have used this before, but just in case, here's a quick little review of how a spectrophotometer works.
Spectrophotometer demonstration. The purpose of a spectrophotometer is to measure the amount of a certain wavelength of light that is absorbed by an aqueous solution. The amount of light that is absorbed depends on the materials in the solution. Seen here are some common buttons on spectrophotometers. The wavelength of light, currently set at 580 nanometers, can be adjusted with the arrows.
Hey, wake up. This is important.
Prior to taking sample readings, the spectrophotometer must be calibrated. In some cases, a cuvette filled with sterile water or a blank cuvette can be used. After placing the cuvette in the holder, close the lid and press the calibrate button. Do not remove the cuvette until the absorption reads 0. Once properly calibrated, samples may be placed into the spectrophotometer and the absorbance recorded.
Good Lord, that was boring. Anyway, what's the next step? All right, now that our sample's prepared, we've added the Drabkin's reagent to it, and we're ready to put it in the spectrophotometer vial and then read it on the spectrophotometer. Okay, so here's our sample, and we take it, and here it's a spectrophotometer, and the sample goes in the spectrophotometer slot, and of course then we turn the dial to the desired wavelength, and we can read our sample. Right? And that's all there is to it for the spectrophotometer. All right, anyone have any questions? No questions? No questions?
I have a question.
You're not even in this class. So now we've measured hemoglobin and hematocrit in a number of different animal species. Now we can take those data, run the numbers, do some additional analyses based on those numbers, and then your assignment will be to write all this up in terms of what do these blood parameters tell us about the lifestyle of the animals.